Hey there. So today we're going to take a look at our notes on transformation and transgenic organisms. So this is going to be packet pages 94 and 90 and 99. So this is going to take up 2 pages in our notebook. I have already cut out and glued in my notebook pages so that I can go ahead and get started on my notes. So first thing we need to do is talk about what is transformation. Last time we talked about how to use restriction enzymes to run a gel electrophoresis and get a DNA fingerprint, but that's not the only thing that restriction enzymes can be used for. So transformation is the process of putting a gene for a special protein from one organism into bacteria so that you can get the bacteria to make that special protein all by itself. We can now call the bacteria transgenic. So transformation is the process of putting a gene from one organism into another organism. And typically, the transgenic organisms that we're making are bacteria. So this transgenic, right, is a living thing that you've added new DNA to. So transformation is the process of adding a gene from one organism to bacteria to create transgenic organisms. And again, typically those organisms are bacteria, but we will look at a couple of different examples as well. So this word transgenic, right? Genic kind of meaning gene, and trans like transfer. So we are transferring genes from one organism to another. So let's look at an example of how we can use transformation and creating GMOs, genetically modified organisms, right, for the benefit of humans. So, for example, let's put the human gene for insulin into bacteria so that the bacteria will make insulin for you. Now, not you like individually, but like you as in like as humans. So if you suffer or you know someone who suffers from who is diabetic, they need insulin. And so we have figured out a way to mass produce insulin for people that suffer from especially type 1 diabetes. So step 1, we need to cut the human gene for insulin out using a restriction enzyme. Remember, we talked about how restriction enzymes are basically DNA scissors. So we can cut out, right, the gene for human insulin, right here. Then, step 2, we will cut the bacterial DNA with the same restriction enzyme. We will then mix the insulin gene with the bacterial DNA. Their sticky ends will match. So because we use the same restriction enzyme, we got the same type of end, either sticky or blunt, so that they would match up together like a puzzle piece. So here was our human gene for insulin, right? And here is the, right, bacteria. So we would just, plop in, right, the human gene for insulin into the bacteria. So then step 4, we put the recombinant DNA into a bacterial cell, right? So here's our recombinant DNA, and it's recombinant because it contains both bacterial and human DNA. So we're going to put it right back into the bacteria. If it works correctly or properly, the bacteria cell should now make human insulin. And this is really incredible because remember, bacteria reproduce very quickly hundreds and hundreds of times a minute. So we are able to produce a lot of insulin that we can, essentially harvest from these bacteria as they reproduce. So, and that's a huge medical and scientific breakthrough when it comes to the medicine and the medical field. Some other benefits of transgenic organisms. So remember, if a living thing has new DNA, we call that organism transgenic, right? Gene and then transfer. So we're transferring genes from one organism to another. We can use transgenic organisms to treat diseases, right? Human diseases like diabetes. But we can also use transgenic organisms to improve agriculture. So here's a couple of examples where we create transgenic organisms that are not bacteria. So we can add a gene for a bug-killing bacteria to a plant. The transgenic plant will kill any bugs that try to eat it. So basically, instead of having to spray a pesticide, which could then run off into the river system and cause pollution, we can create a transgenic plant that is naturally resistant to pests, bugs, and now we don't have to spray. So that's one example of a transgenic plant. Scientists have also cut out an antifreeze gene from Arctic fish and added it to strawberries. This enables the strawberries to grow when it's cold, right? Strawberries they're summer foods, right? You can only get them when it's warm out. But by creating transgenic strawberries or genetically modified strawberries, we can now get strawberries any time of the year because they're now more resistant to the cold and they can grow in colder weather than maybe strawberries that weren't genetically modified, right? That could only be grown in warmer months or in warmer regions of the world. So we're able to expand the food supply, not just for strawberries, but also here, with this plant, maybe this is wheat or rice or corn, a staple crop that millions of people rely on for their food supply. So creating genetically modified organisms, right? GMOs, right, can help increase the food supply, but can also help cure different human diseases. So that's our first set of notes. Let's take a look at this transformation process in a little more detail. So these are our notes. I believe this is packet page 99, and then you should have packet page 90, which I've cut out here, and I'm going to use these phrases to fill out this graphic organizer. So I'm just going to set this aside so I can see it. I only need to glue this page right here into my notebook. So let's start up here, top left. So this is a person that does not suffer from diabetes, right? So they are normal. Give them a little smiley face, right? They do not suffer from diabetes, so they are able to produce insulin normally. So what I can do is I can get the gene for insulin from this person and use it, right? Put it into a bacterium and use it to create more insulin. So here is, right, the human DNA, and this part right here is our gene, right, for insulin. So we will use a restriction enzyme to cut out the gene. Right? And so we've just cut it. We've created blunt ends. Then, right, from the bacteria, the plasmid is removed from the bacterium, and it is cut open, and the human gene is inserted. So this gene would get placed right here, right like that. So I just drew my little pink to represent, right, here's the gene for insulin, right? And I've just plopped it right there inside, right, the plasmid or the DNA from my bacteria. So the bacterium is now transgenic because it contains human DNA, right? So right here, this would be my gene for insulin. So recombinant DNA, right, which is the human and bacteria DNA. So the recombinant DNA is inserted back into the bacteria, right? And it is now a transgenic organism because it contains a human gene and bacteria genes. So now the transgenic bacterium reproduces and copies the human gene from insulin each time it divides. So, right, we go from 1 copy to 2, from 2 to 4, 4 to 8, right, so on and so on, until we have mass produced, right, insulin is produced on a mass scale for diabetics. Right? And then we can bottle it up. You can go to your local pharmacist and get a bottle of insulin. So very cheap, not necessarily easy to make. It does take a lot of hard work to insert that, to cut out and insert the human gene for insulin into the bacterium. But again, if we're successful, if we reach this stage where the bacteria are able to reproduce, right, and copy that insulin gene, then it is very easy to mass produce, right, some insulin for people that are diabetic. So our last question down here, why do you think bacteria are used in the process of producing human insulin? Well, for one, they reproduce asexually, right? So remember, that means that they don't have to find a partner and they can reproduce very quickly. So they reproduce asexually, right? So it's fast, and you don't have to you don't have to go and wait for the bacteria to find someone to reproduce with. So it's very easy to get a hold of a lot of insulin if we use bacteria. So that's it for our notes on transformation and recombinant DNA technology. If you have any questions or concerns about these notes, please just let me know. Okay, I'll see you next time.